Hi, everyone. My name is Easton Farrell. I am a senior in the University of Michigan Honors College of Engineering, and I'm excited to get to present to you some of my capstone findings this year. So I have a very long and complicated title, but needless to say, it boils down to manipulating the environment that cells are in order to favorably control their behavior. So for some background, your body has a lot of different cell types. There's neurons and skin cells and muscle cells, etc. And the way that the body produces these different types of cells is by a series of chemical and mechanical cues that are originated in the extracellular matrix of the cells. Biological research likes to take advantage of this modulative capability of cells by constructing what are called hydrogels. Hydrogel is a small compartment that cells can be implanted into, and you're able to control the chemical and mechanical constituencies and characteristics of the hydrogels. So I have been a part of the Fabili lab since I was a freshman in the Europe program. And the Fabili lab utilizes a specialized type of hydrogels called acoustically responsive scaffolds. And what makes these special is that you can change their properties when you hit them with focused ultrasound. Usually hydrogels are sort of a one and done type thing. Whatever you make, that's just what you're going to be stuck with. But at the Fabili lab, we're able to use this ultrasound technique to change the mechanical and chemical properties with time. The mechanism by which this happens is that we have these embedded entities called phase shift emulsions. And when you hit them with ultrasound, they vaporize and release their innards to the surroundings or expand into bubbles and cause some sort of change. As you can see in these images here, some of the bubbles that form when we hit the emulsions with ultrasound. There's a stable bubble here and then some smaller recondensed collapsed bubbles in the top image. But the important thing here is this halo effect that you see. And that's indicating that there is an increased stiffness of the matrix at the peripheries of these bubbles. And I thought that was very interesting because it's well documented that fibroblast cells can differentiate into myofibroblast cells when they're in a certain high stiffness environment. So I thought that we could maybe, wield this power to cause this differentiation into myofibroblasts to happen. And that would be very therapeutically relevant because myofibroblasts are very critical in wound closure and wound healing. So if it worked, we'd be able to very tightly control a process to generate cells that could heal. So I have here in this diagram just an image of what a typical hydrogel setup would be. This kind of like blue checkered pattern represents the fibrin base for the gel. And you can hopefully see, I'll zoom in a little bit more. There's some blue punctations, blue little dots, and those represent the embedded emulsions. So I would generate these scaffolds, hit them with ultrasound to cause the emulsions to expand into bubbles, and then layer NHDF, which are fibroblasts, on top of the scaffolds. And sort of the hypothesis that I had was the cells that come into contact with expanding bubbles would likely be influenced into this myofibroblast fate because they would be in a higher stiffness environment. So I ran three kind of different iterations of this experiment. In the first one, I put perfluorohexane inside of the emulsion bubbles. I hit the scaffolds with ultrasounds and then layered the cells on top, waited a few days, and then stained for myofibroblast markers. The second experiment sort of accounted for a possibly pernicious effect that would have been generated by the culturing of the cells. The cells are cultured in hard-bottomed flasks, and that's obviously a very high stiffness environment when compared to a gel. So it's possible that just maintaining these cells, keeping them alive in these flasks would have been influencing a fate towards myofibroblast behavior. So what we did with this was we made the scaffolds still using perfluorohexane or C6. But before we exposed to ultrasound, we layered the cells on top and left them in the incubator for 2 days. And the goal of that was to sort of allow the cells some time to renounce the myofibroblast fate and go back to a fibroblast lineage because they're on top of a less stiff environment. And then after that incubation period, we then exposed them to ultrasound. And in the third experiment, we just swapped perfluorohexane or C6 with perfluoroheptane or C7. We did that because the C6, we were having some difficulties with it sort of turning into bubbles before we wanted it to. This happened in this like 2-day incubation period in the incubator, possibly due to the high temperature of the incubator. But C7 is known to have some different properties that sort of ameliorated this effect. So that's why we used the C7s. I'm going to move over to the results here. Zoom out. You can see here a very representative triplicate of images that I would get by looking at the bubble events. If you notice, there's a very significant haloing effect around these stable bubbles and these kind of recollapsed bubbles. They're both formed with the ultrasound exposure being applied to the emulsions. It's just sometimes they become stable, sometimes they don't. But the neat thing here is that we're seeing a higher intensity of this alpha smooth muscle actin signal at the peripheries. The alpha smooth muscle actin is a signal that's specific to the myofibroblast phase. So this was very hopeful. This is indicating that our hypotheses may have been correct, that being nearby some of these expanding bubbles would influence fibroblasts towards the myofibroblast fate. And you can see this sort of like, spooky coincidental similarity between the high stiffness that you can see around the peripheries of the bubbles and the high ASMA signal around the peripheries of the bubbles. So that was very interesting. And we wanted to quantify this so we weren't just presenting images and expecting people to believe us. So the way that I quantified it was by measuring the average pixel intensity of alpha smooth muscle actin normalized per cell at areas proximal to the bubble events. So I drew like a representative region that I would have used and compared that to the average pixel intensity per cell distal to the event. So in areas where we didn't expect to see any differentiation happening. And from that analysis, we yielded these results. If it'll move, there you go. So the important takeaways from this plot are the differences between the pink and yellow rectangles and this darker gray and lighter gray rectangle. So the pink rectangle represents the ASMA signal proximal to the bubble events for the first experiment, and the yellow represents the average signal intensity distal from the event. And there was a very significant difference between those two. Being near the event had a significant enhancement of the ASMA signal and therefore myofibroblast activity. I believe the p-value for this was 0.0007, so a very significant finding. And even in experiment 2, where we were sort of, being hard on ourselves, being cruel on ourselves, and giving the 2 days buffer period, the finding was still significant. There was still an enhancement of the myofibroblast signal proximal as compared to distal. This p-value was about 0.0478. And here is just a nice sort of visual representation that I like to show. This is a line plot that records the average pixel intensity as it passes through the image. You can see here there's some peaks corresponding to the edges of these bubble events. So this recondensed bubble, there's a peak represented here, correlated to the high alpha smooth muscle actin signal you're seeing right there. And then when it hits another bubble, you see another significant peak. So this is just a very nice qualitative way of describing what was happening. And all this led to the conclusion that myofibroblast differentiation, at least as it's reported by alpha smooth muscle actin signal, is enhanced near the peripheries of these emulsion bubble events. And it seems to be that the ultrasound-induced droplet expansion is what's responsible for this effect. So this is very exciting. If we can, get more robust data on this, then we could be looking at a possible therapeutic method. You're able to maybe implant these, expose the hydrogels to ultrasounds, heal, hemorrhaging, internal lesions, things like that. You could also apply it to other types of cell lineages. So you could use some of this data to possibly differentiate mesenchymal stem cells into cartilage fate. That's something that would be very valuable for arthritis treatment. So we'd like to definitely do more research here, but I'm very excited of what we found. Yeah, and thank you for listening to my presentation. Let me find the end record here. All right, bye guys.